Welcome. We are now in paper 3, question 2 from the Kedah state. In question 2 of paper 3, you will typically be given a very short statement or a description, and then you will be required to design a specific experiment, and you are given 17 marks for this question. Usually, following aspects will be required for this question. So I will briefly go through what are the criteria that are required to write for each and every one of these aspects. We will have a look on first the problem statement, and then the hypothesis, the variables, the apparatus and materials, the procedure, and finally the presentation of data. So each and every one of these carries about 3 marks. And it's important that you know the correct technique and to know what are the criteria that are required for each and every one of these aspects. So first of all, for the problem statement, you can and you should always use and modify the questions to make it into your problem statement. I will go through this in further more details when we have a look at the question. And the second thing to remember about problem statement is that your problem statement must always have a relationship between manipulated and the responding variable. And also, the problem statement has to always be in the form of a question, whereby you begin with a question such as why, what, and always end it with a question mark. So if you have all of these 3 criteria, you should be able to have a full mark for this part here. The next is the hypothesis, and again in the hypothesis you need to relate the manipulated and the responding variable. And in the hypothesis, it is also the time where you should predict and state what are the possible outcomes of this experiment, and it is also perfectly fine if you have the wrong hypothesis. You will still be given a mark as long as you show what is the relationship between the manipulated and the responding variables. The next is the 3 different variables. So you have your manipulated variable, your responding variable, and also your control variables or fixed variables. And in this case, you are often allowed to put more than one fixed variable for this section. The next part is the apparatus and materials. So here you will have to list all the apparatus that you think will be used in the experiment. So apparatus refers to usually glasswares, equipments, and hardwares, whereas the materials normally refers to solution, chemicals, and reagents that are used in the experiment. And for the procedure, you are also given 3 marks. And this part here, you are required to show how the results are obtained. And for this part, you can use short sentences whereby one sentence have 1 point. And it's important to also include the following subsections in the procedures. They are classified as K1 to K5. So they deal with number 1, the setup of apparatus. 2, how you handle the control variable. 3, how you control how you handle the manipulated variable. 4, how you handle the responding variable. And 5, what are the precaution or precautionary steps that are taken throughout this experiment. And the last section is the presentation of data. Usually this carries about 2 marks. The way to do this is you should always present your data in a table, and this table should always include title and units. It must also include the title for the manipulated variable and the responding variable, as well as additional calculations that are required for the experiment. And you are not required to fill in all of the blanks. However, the blanks for the manipulated variable should always be filled in. So now that we have gone through all of these sections, let us look at the actual answers and how we can answer this section. Let's just look at the question here. So here they are saying that they have 2 situations, whereby in situation A, a housewife used the product detergent A, and then she finds that it is not easy to remove the blood and grease. However, another person, another housewife, used a product detergent B and to wash her clothes, and then she find that all the stains, the blood stains and the grease are easily removed. So from this, what can we say? So first of all, the active ingredient for detergent A contains 10% of the protease enzyme. So what does the protease enzyme do? It removes, digests the protein. And over here in the chemical, the ingredient for detergent B, it contains 30% protease as well as 10% lipase. So this explains why it is so efficient. Lipase, of course, get rid of the grease, which is a form of lipid. And because here in B you have higher concentration, so it's 30% of protease compared to A, which is 10%. So this is removing the blood, which is a protein. So it seems that from this diagram, one inference that you could draw from here would be higher concentration of enzyme improves the enzymatic activity. Okay, so the next part is they were asking you to design a lab experiment to study the effect of enzyme concentration on the activity of salivary amylase. So it's not the same enzyme, however they are still having the same concept, whereby probably if you have higher concentration of amylase, the activity would be higher. So let us look at how we address each and every one of these aspects here. So the first one is the problem statement. So from the above, we can modify the question that they ask us into something that looks like this. Always start with the question. So here we use what. What is the effect of enzyme concentration on the activity of salivary amylase on starch? So here you are fulfilling the first criteria, whereby you have this in the form of a question. Secondly, you connect the manipulated variable here in green with the responding variable here shown in orange. So as long as you have this 2 plus this statement in the form of a question, you should be able to score marks for this part here. Secondly, the hypothesis. So the hypothesis, again, you need to relate or to state what is the relationship between your manipulated variable and your responding variable. So here you say that the higher the concentration of the enzyme, the higher the rate of the enzyme reaction. So higher and higher, this is the relationship. So this can be totally wrong, you could predict it wrongly. This is also fine, because hypothesis is something that you would like to test in an experiment. So variables, this we already see before. So manipulated variable would be the different enzyme concentration. Your responding variable will be the rate of salivary amylase reaction, and the control variable would be something that you keep constant. So you keep the same volume of starch suspension, same volume of enzyme, and you also use the same temperature for all of the reactions. Apparatus and materials. For the apparatus, you will require a beaker, a water bath, thermometer, test tube, syringe, droppers, glass rod, stopwatch, and white towel. So remember, for this experiment, white towel is very important, as well as the stopwatch, the test tubes, or the water bath. So these are the important items that you need to conduct this experiment. As for the materials, all of these are equally important, so you definitely need your starch suspension, saliva suspension, iodine solution, ice cubes, and also distilled water. As for the procedure, you can see here I have already labeled them. What are the criteria for each of the steps, so you know if you have already fulfilled. K1, you will require at least a few 2 to 3 of K1s, and at least one of the other K2, K3, K4, and K5. The way you do this is, first you set up your apparatus. So this will be K1, so you need to label always your tubes. So here you can use 6 tubes, you label it differently: A1, A2, B1, B2, C1, C2. And afterwards you will need to collect the saliva. So this you collect it using a beaker. This is still the experimental setup step. And then afterwards you dilute it with distilled water into 10% saliva or amylase suspension in test tubes A1. And then the next thing is you need to fill up another test tube, which is called A2, with 2 ml of saliva using a syringe. Afterwards, both test tubes are supposed to be immersed in a beaker containing ice cube for 5 minutes. So this is to make sure that both of the test tubes starts at the same temperature. And while waiting for this, you can add a drop of iodine solution into the groove of a white tile. Next, 5 ml of starch suspension is added to the test tube containing saliva and stirred using a glass rod. The stopwatch is then immediately started, and a drop of the mixture from the test tube is then placed to a groove on the tile containing the iodine solution. Afterwards, the iodine test is repeated for every 30 seconds until the mixture turns brownish yellow. So this is the time when the starch, which is originally blue-black solution, will then turn into brownish yellow. So this is where the starch is no longer starch, which means it has completely broken down by the amylase enzyme. So next, the time taken for the iodine solution to turn brownish yellow is then recorded. The rate of enzyme reaction is calculated using the formula. Rate of reaction equals to 1 divided by the time taken for the iodine solution to turn brownish yellow. So this step of having the formula is very important because you will require to calculate this rate or anything like that for this kind of experiment. So it's important to always include the formula that is relevant for the experiment. And following that, step 3 to 12 will be repeated using 15 and 20% of saliva suspension, and then afterwards all the data is recorded in a table. The experiment is then repeated twice to get the average reading, and then you can plot a graph of the rate of enzyme reaction against the enzyme concentration. So it's important to note that after the first part, it is always essential to state that you are repeating the rest of the experiments again using different manipulated variable. Finally, for the presentation of data, you always need to draw a table, and it's always important to have at least your manipulated variable as well as your responding variable and the calculation. So here you will have the concentration of amylase, which is 10%, 15% and 20%, and the rest of this you do not have to fill up. So with that, this is the structure and technique how you can answer the second question of paper 3.